This is a video overview of energy and metabolism. Recall, all living organisms require energy. For an organism, energy is necessary to do work. In terms of a cell, work can be constituted as chemical work, mechanical work, or transport work. So when you think about energy and their requirements that cells have, we can think of these in terms of metabolic reactions. Metabolism is all of the reactions involved in a cell. And metabolic reactions are regulated through stepwise pathways. These metabolic pathways can be divided into two categories: anabolic pathways and catabolic pathways. Metabolic pathways involve the transformation of energy. And the study of the transformation of energy is referred to as thermodynamics. So we must think of energy in terms of the fact that we're in an open system. And in biology, we're interested specifically in two laws of thermodynamics. The first law is the conservation of energy. It can neither be created nor destroyed, only transformed. The second law of thermodynamics is that entropy will always increase. So if you think of a cell, it's an open system, and its environment is its surroundings. According to the first law of energy, of thermodynamics, a change in energy in the system must result in a corresponding change of energy in its surroundings. The system will also spontaneously evolve towards thermal equilibrium, which means there will always be an increase in the randomness, according to the second law of thermodynamics. That means for living organisms, we must constantly be inputting energy into the system, or disorder will continually increase. This is also why spontaneous reactions will happen. A non-spontaneous reaction leads to a decrease in entropy, and that will only happen if energy is supplied into the reaction. So we have this open system, and again, it's important to understand that we can measure the amount of energy needed for the system. This is the free energy. It's G, known for Gibbs free energy. That's the portion of the system's energy that can perform work. The change in free energy, or delta G, from a biological perspective, is that delta G will be positive because the system gains energy. However, if a reaction is exothermic, also known as a spontaneous reaction, it will lose energy, and thus delta G will be negative. Cellular respiration is an exothermic reaction. So when glucose reacts with oxygen, the products of carbon dioxide and water are formed along with potential energy. This potential energy is released, and the delta G is negative. In the case of exothermic reaction of cellular respiration, the new product will become ATP, adenosine triphosphate. ATP molecules are high energy molecules. That's because the phosphate bonds have negative oxygens associated with them, and these are pretty unstable. They want to repel away from each other. ATP is hydrolyzed to form ADP plus inorganic phosphate and energy. In a cell, pathways are going to harness the energy to drive other reactions. We call this coupling reactions. So the hydrolysis of ATP will be accompanied by the phosphorylation, or addition, of a phosphate to another molecule, allowing for work to be done. The example of a coupled reaction of glutamate synthesis with ATP hydrolysis in your textbook illustrates how taking that exergonic reaction of the breakdown of ATP and coupling it with the endothermic reaction of creating glutamate allows for an overall spontaneous reaction to occur, and the delta G is negative. So the cells do this continually in order to synthesize new products and hold back the ever progression of entropy. Even though a reaction is spontaneous, the time it takes for the reaction to proceed forward can fluctuate and really can be very slow. Life needs things fast, so enzymes are the answer to this dilemma. They allow reactions to go forward much quicker than normal. That is because enzymes are catalysts, special proteins that speed up the rate of a reaction without being used up. How do enzymes work? They work by lowering the energy activation barrier. Every reaction has an energy activation barrier. So what is necessary is to be able to get over that for the reactants to be converted to products. Enzymes lower this barrier. Each reaction has its own separate enzyme, and this works because of substrate specificity. We can think of the reactants as substrates, and these substrates bind to a site on the enzyme called an active site. So what we can see here is that the enzyme binds with the substrate, forming an enzyme-substrate complex. While the enzyme and substrate are joined, the catalytic action of the enzyme converts the substrate to a product. In this fashion, and because enzymes never are completely used up, only a very small amount of enzyme is needed. How do they perform this cool function of lowering the energy activation barrier? They do it in 3 different ways. They can basically provide an active site that provides a template in which substrates can come together and in the proper orientation. Or the active site might provide a nice microenvironment conducive to the reaction. Or the amino acids in the active site might directly participate with the reaction. The point is that without these enzymes, the reactants would not be able to have the right conditions to go forward in a short amount of time. Keep in mind, enzymes do not change the amount of free energy, they just speed up the reaction rate. And because enzymes are reusable and they do such good jobs, they do need to be regulated. And so there are different types of inhibitors and activators that we talk about. So again, there are competitive inhibitors, non-competitive inhibitors, allosteric inhibitors, and allosteric activators. There's also feedback inhibition. So in summary, metabolism is all the chemical reactions that occur in a system. And again, these reactions happen in a controlled pathway way, right? So each step is regulated. And to go from one step to another in a timely fashion, enzymes are necessary because they act as catalysts. Hope this helps you understand a little bit better energy and metabolism in a cell.